Good afternoon, everybody. Welcome to session 2 of our virtual symposium. That was a great lead up there, Scott, setting the stage for us with the urchins and what's going on the nearshore rocky reefs in Oregon. So we'll be getting to our second panelist who will help to expand on some of those topics. So I'm going to share my screen here and say thanks again for attending. There may be a few of you who are now coming on who hadn't been here for the first session. I want to again thank our co-sponsors, the Hatfield Marine Science Center, Oregon chapter of the Wildlife Society, and also the Meyer Memorial Trust for financial support for the Elakha Alliance. You can check us out at elakhaalliance.org. Google has come to recognize that. So for the participants, please mute yourself during the webinar and send questions to John either in the chat room or the Q&A online. And we'll run this particular session like we did the last one to the 55-minute mark, and then we'll take another 5-minute break. So I want to start the second session here by introducing our next speaker, Sara Hamilton, and a little background. So sea otters, as we all know, are a predator of sea urchins, which, if unchecked, will devour kelp forests. Sea stars, and especially the sunflower sea star, the Pycnopodia, as Scott said, are also voracious predators of sea urchins. So for more than 150 years after sea otters were exterminated from the Oregon coast, the sunflower sea star provided an ecological backstop to protect kelp beds. But a wasting disease in 2013 wiped out most of the Pycnopodia and other sea stars along the west coast. And as a consequence of this and other factors, the population of sea urchins exploded and kelp beds rapidly began to disappear, including on the Oregon coast. So as we think about restoring ecological health to our nearshore ocean, this requires us to understand sea stars as well as sea otters. And our next speaker, Sara, will help us do that. Sara is a PhD student and an NSF GRFP fellow at Oregon State University. She did her undergraduate degree in biology and gender studies at Bowdoin College in beautiful Maine. Her PhD thesis work on bull kelp forests takes an applied and interdisciplinary approach, examining many parts of the kelp forest systems, including spatial ecology of sea stars, especially the Pycnopodia sunflower sea star, the population dynamics of bull kelp, diets of purple urchins, and best practices in kelp forest conservation. She also works with partners such as the Nature Conservancy and the Oregon Kelp Alliance to support kelp forest monitoring, science, and decision-making. She spoke to on kelp resources at our science symposium in October of 2018 and has a very informative podcast that is posted on the Elakha Alliance website. However, for this talk, she has shifted her focus to include the status of Pycnopodia and its role in nearshore marine ecosystems. We are pleased to have her share her research with us. Sara, it's all yours. You may share screen.
Thank you, Bob.
You bet.
Let's see. Here we go. Is that up?
That's up.
Wonderful. Let me get all set up here. Okay, thanks for that introduction, Bob. Hi, everyone. My name is Sara Hamilton. As Bob mentioned, I'm a PhD student at Oregon State University. And I know this is a talk about, or a symposium about sea otters, but I'm always one of the people they invite in to talk about things other than sea otters. I'm a kelp forest ecologist, and I'm here today to help us think a little bit more about another top marine predator in Oregon that we've lost, which, as has been alluded to here, is the sunflower star, also known as Pycnopodia. I also want to note that I'm here today speaking on behalf of not just myself, but the Pycnopodia recovery team, which is a large group of folks that have been working together to combine data on Pycnopodia, better understand how their populations are doing, and explore options for conservation. So I just wanted to point out that this presentation is the product of a large multinational team effort. And before I get started here, I wanted to mention two other things. The first of which is I want to acknowledge the fact that as I give this talk, I'm living on the traditional lands of the Mpinafu band of the Kalapuya peoples. The Kalapuya peoples were forcibly removed from these lands in the 1800s, and the living descendants of these people are today members of the Confederated Tribes of Grand Ronde Community of Oregon and the Confederated Tribes of the Siletz Indians. I'd also really like to thank the representatives of some of the Oregon tribes who helped start the Elakha Alliance and have poured a lot of time and energy into this effort, as well as thanking Bob and John for all the work they've been doing with the Elakha Alliance and to set up the symposium in particular. And also, I want to just make the audience aware of one thing, which is actually some of this information that I'm going to be sharing with you today isn't quite yet published. And because one of the things that we're trying to do is raise awareness around Pycnopodia's status and what conservation options we should use moving forward, we are hoping that once it is published, it will get some relatively high profile media coverage. And so something I've learned from our media team is that if smaller publications get to start publishing on a story first, larger, more high profile, like national level media publications are less likely to pick it up and run it. And so I'm going to be a little cagey with some of the information here today. I'm not going to be able to share exact numbers because we're trying to wait until some of this work is officially published so we can get the kind of media splash that will create the most awareness and best outcomes for Pycnopodia. So I apologize for that. If I'm a little cagey with some of my numbers, that's why. So thank you for bearing with me here, and let's dive in. So just to give you an idea of what we're going to be talking about today, I'm going to introduce you to the sunflower star. I'm going to happen to them, why they're pretty much all gone now. Talk about where are they now, discuss conservation options for the future, and then kind of talk about what does this mean for Oregon, particularly in the context of these conversations around otter reintroduction. And if that popped on my screen, yes, it did. So I've been trying to use the common name of sunflower star because a lot of audiences have an easier time with common names. However, the scientific name Pycnopodia is the one I always use, and I'm so used to using it that it just kind of rolls off of my tongue. So if I'm switching back and forth, just to be clear that when I say sunflower star, I also mean Pycnopodia and vice versa. So let's introduce So what is the sunflower star and why is it worth talking about? So actually, the sunflower star is the largest sea star in the world, and it can grow to be over 3.5 feet across. So not the small intertidal sea stars you see. These guys can be bigger than dinner plates. They can be quite massive. They're also one of the world's fastest moving sea stars. They can grow to have more than 20 arms, up to like 26, 27, and are voracious predators. They are generalist predators, which means that they can eat a lot of different things, but some of their favorite foods are clams, urchins, mussels, and snails. Internally, we kind of refer to them as like the grizzly bears of the sea because they are top predators, but they're very general in their diet, very general in what they eat, and can almost act like scavengers in some circumstances. Historically, they range from Baja California all the way down in Mexico up to the Aleutian Islands in Alaska, which you can see here on this map to the right. Historically, they tended to prefer depths down to about 200 feet, so relatively shallow, but they've also been found at depths exceeding 1,000 feet, so they can go deep as well. Historically, they were pretty common across most of this range and were a regular occurrence for divers diving along the West Coast. So as voracious predators, sea stars can have a big impact on the food web and associated ecosystems. And since Pycnopodia do indeed prey on urchins, there's the potential that the sunflower star can promote abundant kelp forests by keeping urchin populations low. And there have been several studies that have found that when Pycnopodia were removed from a kelp forest ecosystem, there tended to be more urchins. The urchins were out and about grazing on the reef, and consequently, there was less kelp. 2 of those studies came out of British Columbia, and one of them came out of Southern California. So we know that they have the potential to impact the state and the resilience of kelp forests. However, there are a lot of things that we don't understand about the role that Pycnopodia play. So some of the questions I have up here that we have yet to understand is if there are other predators in the system, like otters or maybe sheephead fish, does Pycnopodia influence urchin densities as much? If the urchins are starved, like Scott was talking about with, they have, their roe is much less rich and nutritious. And so the urchins are starved as they are in a lot of barrens, will Pycnopodia still eat them? If Pycnopodia have access to lots of other prey, lots of clams and mussels or snails, will they eat as many urchins? Will they exert that same kind of top-down pressure on urchin populations? And there are a lot of other questions. So we need a lot more research, both in the lab and in the field, to better describe and predict how the presence of Pycnopodia influences kelp forests. But one thing that I want to point out here in the context of this being a sea otter symposium is I want to talk a little bit about predator redundancy, which is a term ecologists use to talk about how many different species of predators are there in a system. If you look at this map that I've got over here to the right, I've laid out the ranges along the west coast of some of our major urchin predators. So we've got Pycnopodia's range in purple there in the background, kind of. I laid my best understanding of Otter's current range in brown. Sheephead fish, their range is in dark, and spiny lobsters are in red. If you're unfamiliar with any of these species, that's fine. Just know that these are all major predators that can eat or scare away urchins and have been found to promote kelp forest health through their top-down control, their predation on urchins. So kelp forests are present across this entire swath of the West Coast, from Baja all the way up into the Arctic. And so I just kind of want to point out that there is a good chunk of the range of kelp forests on the West Coast in which Pycnopodia are really the only major urchin predator, or one of maybe only two. In places where you don't have otters or sheepshead or lobsters, Pycnopodia is one of the only predators that we're aware of that can really eat urchins in high enough quantities to potentially reduce their densities in a way that's meaningful for kelp forests. So there are a lot of places along the West Coast that, with the disappearance of Pycnopodia, which I'm going to talk about in a minute, there essentially aren't any major urchin predators left, and Oregon is one of those places. So those were the really cool things about Pycnopodia, and now we're going to get into the not so cool part. Many of you are probably at some point have heard about sea star wasting syndrome, which was a massive epidemic that broke out around 2013 and started killing sea stars up and down the coast. Over the course of the next couple of years, it struck essentially the entire West Coast, from Baja, Mexico to Alaska, affecting multiple sea star species and dramatically reducing their populations. Up here on this right picture, you can see a picture of a wasting Pycnopodia, and Scott kind of mentioned earlier how they melted away. Well, you can see it does kind of look like they're melting almost, or like they're decomposing, and that's what wasting does to these critters. And to try to give you an idea of just how quickly this happens in some places, I want to kind of point you over here on the bottom. This is a set of pictures that came out of a publication by Drew Harvell, and they were taken in the Salish Sea, so Puget Sound area roughly. And what you see on the left is in early October 2013, this rock was completely covered in Pycnopodia, almost you can barely see the rock underneath. And then when the same diver went back just a few weeks later, that rock was completely barren, and he didn't see a single Pycnopodia on his dive. So the speed at which this happened was truly stunning in certain places. And this is this kind of wide-scale sudden loss is an observation that many people all up and down the coast were making. So just how bad was this crash? And this is where I have to get a little cagey because things aren't quite published yet. But my team, the Pycnopodia recovery team, a team that's funded by the Nature Conservancy, undertook an IUCN Red List assessment. Now, IUCN stands for the International Union for the Conservation of Nature, and they host something that's called the Red List, which is an international designation of whether a species is endangered or not. So basically, what these Red Lists assessments require is that you do an assessment for the entire species, not just a species in one part or another, but worldwide, how is this species doing? And so what we needed to do was go in and assess from Baja to Alaska how much their populations had been reduced. And I want to point out that we had 33, it might be even higher now, 34 or 35 contributors that shared over 61,000 surveys with us so that we could try to cover that entire, incredibly thousands of miles of coastline. So I just want to point out that this was an incredible team effort, and we are so grateful to those people that shared data with us, and the cool results that we're getting and we want to share with the world soon are directly because of their efforts and their willingness to work with us. Let's see here, where are my notes? So while I can't give you the exact numbers right now for the global decline, so the total worldwide decline in Pycnopodia population size, I can tell you that we suggested to the IUCN that this species be listed as critically endangered, which is actually the most severe endangered status a species can receive from IUCN without being listed as extinct in the wild or extinct. And one of the bars for being listed as critically endangered means that there's been at least an 80% reduction in population size. Additionally, we estimate that billions of individuals died in this epidemic. So this truly was a tremendous shock to the species, and their populations have been dramatically slashed since this outbreak took place. So where are they now? So using these 61,000 surveys that people have shared with us, it's 4 to 7 years post the real outbreak of this epidemic, depending on where along the West Coast you lived. And what I can tell you without giving us specific numbers again is that there really aren't any Pycnopodia left on the outer coast of the lower 48 states of the United States. We estimated that these populations have declined probably around 98 to 100%. And in fact, it really does look like they're locally extinct across these thousands of miles of coastline. For instance, of the thousands of surveys we looked at in the lower 48, not a single Pycnopodia has been found in Mexico since 2015 and in California since 2018. And ever since the crash happened, only about 24 individuals have been found in Oregon or on the outer coast of Washington. However, that doesn't, I was talking about the lower 48. The one exception is the Puget Sound. As you get to the Puget Sound and then you start to move north into British Columbia, the Salish Sea, you start finding remnant populations again. Overall, we found that in the Salish Sea, British Columbia, and most of Alaska, their populations had probably only declined by something like 90 to 95%. So still an enormous population crash, but the difference is that they still have remnant populations up north. They still sometimes see them on dives. It's still very locally patchy in the northern half of their range. And in some places like Haida Gwaii, they've barely seen any since the outbreak. But I want to show you a cool picture. This picture actually came from some divers that we're in touch with that were working on behalf of the First Nations along the central coast of British Columbia, and they were diving in the fjords along the central coast. And I want to point out that they're a little hard to see in this picture, but this is actually a single picture that had 4 separate Pycnopodia in it. Each one is circled here. And we were getting reports of them finding, 20, 30, 40 or more Pycnopodia in a single dive. So, that's exciting news. And that's something that scientists down here in the lower 48 were really jazzed to hear. I do want to point out that these remnant populations in the northern half of their range appear to be pretty stable, but they are still at incredibly reduced densities. So that's where they are now. Given that it has been several years since this outbreak really first hit, have these populations begun to recover at all? And we've seen recovery in some other species of star. For example, the ochre star, also known as Pyzaster ocraceus. In a lot of places along this range, their populations have been going back up. You can go down to the Oregon coast and spend some time in the intertidal and see them, see them tomorrow. So some star populations are increasing, but based on the data that we've collected, based on all these surveys, we would say that no, we don't actually see evidence for recovery, even in those places that still have remnant populations. Now, I apologize, this graph is a little difficult to see, but the important parts are that along the x-axis, we're looking at time from 2017 to present day, 2020. And along the y-axis, we're looking at the density of Pycnopodia found in different surveys. The various lines represent, the various lines have different colors, and those colors represent each individual region that we looked at. And I know they're a bit difficult to see, but really the only thing that you need to notice about these lines is that none of them are increasing. None of them show populations rebounding in any of these places. On the outer coast of the contiguous US, populations have flatlined around 0, at essentially none. And in the more northern areas, they've remained steady at this kind of dramatically reduced population, or in some places we're seeing small bits of decline over time still. So that's a little, that's a cause for concern. And one thing to note is that in some of these more northern regions, there has been evidence of recruitment events. So recruitment events, I just mean juveniles coming in. So in places like Alaska and British Columbia, people are seeing juveniles, which indicates that some amount of reproduction is still happening. However, whether those juveniles are making it to adulthood or whether maybe they're being knocked off by wasting syndrome is still unknown, essentially. We're still trying to figure that out. Overall, considering that this is likely a relatively long-lived species with a long generation time, and considering their current low population densities and their lack of current recovery, we don't expect to see we're not seeing a major recovery of the species, and we don't expect to see one anytime soon. So just to kind of recap there, what happened to the sunflower stars? There were catastrophic losses globally. And when I say globally, I just mean throughout their entire range. They're nearly extinct on the outer coast of the contiguous US. We find remnant populations dramatically reduced, but existing in the Salish Sea, British Columbia, and Alaska. And we're not seeing any evidence that their populations are rebounding post-outbreak, which is, like I said, a little bit of a cause for concern. So I want to talk a little bit about what do we do now? What are our options if we care about conserving this species? I think the first thing that I really need to point out is that before we do any real conservation, we need to know more about the species and about the disease. I mean, this is a species that historically wasn't harvested, and therefore there's not a lot known about it. There wasn't a lot of research done. A lot of research is just done when there's a species that humans care about because we're harvesting it. I mean, for example, it's been 7 years since this outbreak really started. It was probably one of the biggest disease-related die-offs of animals in history, and we still don't know what the disease is. We literally don't know what pathogen it is that causes these animals to die. We thought it was a densovirus and realized that that might not be right. So, we have to be able to identify the pathogen that's causing this disease before we can do any serious conservation work. And I'll talk a little bit about that in a minute, why. Also, as I mentioned, we just don't know that much about the species itself. And that's something that we discovered while doing the IUCN analysis. IUCN, the Red List assessment, was asking us, how long does this species live? Where does it breed? What season does it breed in? Are there critical habitats that it depends on? And for all of these questions, we just had to say, we don't know. There's no published literature about it. Partially because they're really hard to raise in captivity. They don't like living in captivity. So, and then additionally, another one of the key research gaps that really needs to be answered before some of these things can move forward is if we want to try to use sunflower stars to help promote kelp forest health, we need to better understand how and when Pycnopodia, or the sunflower star, can promote kelp forest growth. I spoke, we still have so many questions about that. And so if we're going to try to use these for kelp forest restoration, we need a better understanding of how that's going to happen and if it's going to happen reliably. So with an understanding that more research is one of the first steps to doing any kind of conservation action, What are some of our other options? Well, these are some of the options for conservation that our sunflower star recovery team came up with. And the first thing that came up was the idea of translocations. Now with translocations, we're talking about moving sunflower stars from the wild, from one place, maybe British Columbia or the Puget Sound, and moving them to a place without sunflower stars. This is how, sea otter reintroductions have historically been done. And currently this is a problem because we don't know what the disease is. And because we don't know what the disease is, we don't know how many of the remaining populations up north may be contaminated with that disease. If we don't know what the pathogen is, if we don't know what virus or bacteria or something we're looking for, we can't test for it. So we could be moving diseased animals across state lines and reintroducing the disease elsewhere. So that's not a very appealing option. It would be a crazy hassle to try to get permits with. So our team didn't have high hopes that that would be a feasible option anytime in the near future. However, a potentially easier or safer option could be to raise non-diseased sunflower stars in captivity and then release them into the wild. Now, I can tell you that there are captive breeding efforts underway in partnership. I think the Nature Conservancy is funding some captive breeding work that's going on between University of Washington and the Seattle Aquarium. But at this time, there currently aren't any plans to try to reintroduce those individuals to the wild. However, if in the future that becomes something that Oregon or Northern California or somewhere else is interested in pursuing, we need to already have these captively bred populations in the lab and established to be able to respond, if people become interested in the idea of restocking or reintroduction in that way. So finally, two other options that we might have for conservation would be to, one, minimize other sources of mortality, and two, look at habitat protections. And so when I say minimize other sources of mortality, I'm particularly talking about the fact that the sunflower star gets caught as bycatch in trap and line fisheries, particularly crab trap fisheries. So we don't know if this is currently impacting the populations. We don't know if getting caught in a trap, getting hauled up and then thrown off deck, we don't know how often that might kill individual sea stars. And it's probably not killing a large number of them, but if we've got these really small, fragile remnant populations in places that are pretty vulnerable, we certainly don't want those individuals from those, fragile populations to be accidentally killed in crab traps or other fisheries. So potentially doing outreach with fishers to talk about the fact that this is an endangered species that may have important effects on the ecosystem, and to talk with them about safe ways to release the any sunflower stars that they find as bycatch. That could remove some extra mortality, some extra pressures from these fragile remnant populations. And then two, we heard about, or I mentioned habitat protections. So there may be some habitats that are particularly important for Pycnopodia. For example, we've heard reports of Pycnopodia juveniles using seagrass meadows as nursery habitat. So if there are habitats that are particularly important to stars, they tend, as adults, they tend to live in most habitats. They're not very, specific to only this area or only that area. But if, particularly for juveniles, if they have important nursery habitats, protecting those habitats could set the species up for successful reproduction and rebounding of their populations. However, as I was saying earlier, we don't actually know that much about what habitats are important for those sunflower stars. We don't know if those habitats are under some kind of, external pressure. So as I mentioned, with this conservation option and with all the others, we'd need to do a lot more research before we were actually able to use it in any kind of informed way. Let's see. Oh, and then one last thing, what can you do? One thing that we could certainly use help with is tracking remnant populations, particularly if you see any juvenile or adult Pycnopodias in the lower 48, particularly less so on the Puget Sound and more so on the outer coast of Washington, down all the way to Mexico. If you do spot any Pycnopodia in the wild, please report it either at seastarwasting.org or in iNaturalist. If you can take a picture, if you can note whether they look like they're wasting or any other details, how big they are, and then report it to seastarwasting.org or the iNaturalist project. That helps us, and we don't have eyes on all the populations all the time. There are lots of people out there looking in the ocean, and so that anecdotal evidence can help uncover a lot, especially when there are such small populations left. So particularly if you go to iNaturalist, you'll have to go and search for, I have it over here, this marine wildlife health project, and you can report any sightings there. So I know I talked a lot kind of about the global picture for Pycnopodia and for the sunflower star. So I want to just kind of bring it home and talk about what does this mean for otters and for Oregon. And I want to start off by saying I don't know what it means. Conservation is a tricky issue that scientists aren't the only ones that are providing insight. Conservation is a lot about community values. And so, things that scientists can't answer. But some things that I do think we want to think about are, as I kind of mentioned, considering how few there are in Oregon now, almost none, and considering how long-lived they are, we're unlikely to see sunflower star populations rebounding in Oregon anytime soon. And then additionally, as Scott set me up really well to talk about, we are seeing increased purple urchin densities, which are one of the species that sunflower stars like to eat. This is actually, I was snorkeling or diving down at Cape Arago not too long ago, and this is an urchin barren that I ran across. And so we are hearing lots of reports of massive increases in purple urchins, whether that's related directly to the loss of sunflower star or new recruitment events, changing water conditions, we don't know. But additionally, as Scott kind of mentioned, we are seeing areas where our kelp forests are declining. And I don't think we necessarily saw the same kind of large scale collapse of the kelp forests post 2014, 2015, that was seen in Northern California. If you've been paying attention at all to the kelp forest collapse in Northern California, we didn't see that same kind of synchronized statewide sudden collapse, like they were all gone. But we are seeing important places that are losing their kelp forests. Scott was talking about Orford Reef. Orford Reef historically was one of the biggest kelp forests in all of Oregon. And it's really depleted these days. I was diving Nellie's Cove down in Port Orford not too long ago, and it's a complete urchin barren right now. And some of the local folks down there were telling me we used to bring people here to go kayak fishing because there was so much kelp, it was great fishing habitat. So we are seeing localized declines of kelp that are probably related to urchin densities. It may not be the only cause, heat waves and lots of other things that are changing in our environment are probably contributing to the problem. But I can tell you that having lots of urchins around probably isn't helping. It's probably making the problem worse. So with that in mind, I just kind of want to emphasize, if we are interested in reintroducing otters because of the potential implications it has for kelp forests, I just want to point out that there is another major urchin predator, the sunflower star, that has disappeared from Oregon. And there are going to be really different trade-offs in trying to reintroduce both of those species. So for example, there are different people that are invested in otters as opposed to sunflower stars. Otters may impact the Dungeness crab fishery, whereas sunflower stars aren't quite fast enough to catch Dungeness crabs most of the time. So sunflower stars might not impact the Dungeness crab fishery. On the other hand, some of the tribes in Oregon have cultural connections to sea otters that are much stronger and more important than cultural connections they had to the sunflower star. So I'm not saying we should focus our energies on one or the other. I just kind of want to remind everyone, you know, otters are so charismatic. I just want to kind of remind everyone that there is another major predator that we've lost and that is potentially ripe for reintroduction. And to just keep that in mind as you move forward and you start thinking about what are the trade-offs, what stakeholders am I going to piss off, what stakeholders am I going to make happy if I try to do this reintroduction. It's just kind of nice to keep that in the back of your head and think about what really restoring the whole kelp forest ecosystem would look like, as opposed to just maybe restoring individual species one at a time. So I tried to keep this relatively short. So I'm gonna wrap up there. Before I do, I wanna have a big shout out, thank you to a lot of people. Particularly I wanna, you know, I talked about the Pycnopodia recovery team. I wanna really point out some of my key collaborators, Sarah Gravem, who is a postdoc at Oregon State University, and then Vienna Sakamano and Walter Hetty, who work for the Nature Conservancy. The Nature Conservancy has been funding a lot of this Pycnopodia recovery team conservation work, and so we really appreciate their support. And then this is a list of all the people who contributed to our IUCN Red List assessment, many of whom shared data with us. And as I mentioned before, you know, we wouldn't be able to say these kind of things about Pycnopodia populations without their cooperation, without their support, without their insights, without their data. So it's been a really cool project to work on, to get to work with so many different people and learn, you know, what the situation's like in Alaska versus the Puget Sound versus Southern California. So I'm so grateful to all of those people. Also, if you have questions, if you're interested in sharing data or what kind of data we have, if you want to join the recovery team, feel free to either email me at hamiltsod@oregonstate.edu, or you can also find me on Twitter. So I'll go ahead and wrap it up there and open it up for questions.
Thank you, Sarah. That was great. And I have a few questions here. The first question is from James. What is the range and preferred size of the Mytilus species consumed by Pycnopodia, I assume he means?
That's a great question. You know, I'm not an expert on mussels. I know that I believe there are two main mussel species on the West Coast. I don't know what their ranges are. And I'm not sure, you know, there is some primary literature that shows spots where Pycnopodia really prefer mussels or Mytilus species. You might be able to find that and see what the range was for the size range that was preferred for mussel prey by Pycnopodia. But to tell you the truth, I'm sorry, I don't know that off the top of my head.
No problem. There's a question from Philip, and it's regarding Pycnopodia. Could wasting be related to, and you've alluded to you alluded to some of this, but could wasting be related to non-biological or non-pathogenic causes like temperature, or other water quality, salinity?
Yep. Yeah. So we know that it does interact with the environment. So there has been some really good studies that have demonstrated that in places where water temperatures are higher, you get worse outbreaks of sea star wasting syndrome, and the Pycnopodia die more quickly. They get worse versions of this wasting syndrome. And so it's very likely that one of the reasons we got this enormous outbreak in 2014 had to do with the fact that we were going through a historic marine heat wave at that time period. If anybody remembers the term the blob, that was this historic marine heat wave that swept much of the West Coast, stuck around for a long time, and increased temperatures, you know, in ways we've never seen before. And so it's likely not a coincidence that that outbreak happened at the same time that we were getting historically warm waters. There are likely other interactions with environmental conditions like water quality or salinity, or maybe even depth. But, you know, without knowing what the disease is, what the pathogen is related to this syndrome, we can't do laboratory tests. And so we're kind of stuck just like trying to figure out, based on where they are now, what environmental conditions enabled greater kind of population level resistance to this disease.
Thanks. Could there, this is from Dave, and could there be multiple species or subspecies of the Pycnopodia that have different levels of immunities?
I don't know about in terms of species. So the species, I was using their genus name, Pycnopodia, but their full name is Pycnopodia helianthoides. So it's a single species. As far as I know, I mean, there hasn't been a lot of research into the genetics and phylogeny of sea stars, but as far as I know, there really is only one species. However, there likely are different, I don't know if I would call them subspecies, but there likely are populations that may have higher resistance to this syndrome. I mean, when you think about, we were trying to do some estimates of how big the population was before the crash, and it was billions of individuals, as I kind of mentioned earlier. I think it was around 8 billion is what we estimated that it used to be. And so if you're looking at 8 billion individuals, you know, when we're talking about evolution, there are likely ones out there that have mutations or have something going on that gives them higher resistance to this disease. It's just a numbers game when you have so many individuals. And so we're not sure if the populations up north maybe had some kind of, the individuals had some kind of resistance to this syndrome, or if it was like an environmental interaction. So like an interaction between temperature and the strength of the disease. So yeah, we're still trying to figure that out. As many of my answers probably will be, considering how little research there, or how little we know about this topic.
Here's a question from Elizabeth, and you mentioned trying to minimize bycatch mortality of Pycnopodia, say in traps, pots. So Elizabeth asked if there's been preliminary outreach, and if so, what has various fishing stakeholders' response been, or attitude been, positive, negative, neutral?
So we haven't gotten to the point where we've been able to try to coordinate any outreach with fishers. I know that historically, based on some interactions I've had with fishers, they tend to view them as a big nuisance. I mean, they're huge, you know, they're big and they're slimy and they're like, they kind of look like monsters, you know, and they get caught in people's traps. They get caught in people's lines. They get caught, you know, pulled up. They don't taste good. Nobody wants to eat them. So I think generally, historically, they've been viewed as a nuisance and kind of like, ugh, another, a lot of people call them rag mop stars because they do kind of just look like a mop. And so, you know, people ripping them off and just throwing them back into the ocean, kind of annoyed. So we haven't done any outreach yet. You know, I would imagine, you know, fishers are smart people. They get they understand ecosystem connections. So I imagine if you did some of that outreach, they would probably be pretty amenable to saying, oh, wow, this is a really endangered species. We'll try to be a little more careful with it. But that is, you know, that would require a lot of time and energy to do that outreach. And unfortunately, right now, we're just really limited on people power, on funding and things like that. But we'd be really interested in collaborating with people who wanted to do some of that.
Thanks. Next question from an anonymous attendee: How have we been able to rule out the denzovirus as the cause of the sea star wasting disease? Evidence previously suggested it was found in specimens from the 70s and maybe 80s that also experience the wasting disease? ?
Yeah, so this is such a complicated question. And to be honest, I am not a disease ecologist, so I won't be able to give you the best level of information there is out there. But based on what I understand, this speaker is entirely right. We have seen episodes of something like wasting syndrome going back for dozens of years. There have been, you know, lots of anecdotal evidence that there were outbreaks, particularly in Southern California and other parts of California in the 60s and the 70s, where, you know, you saw the same kind of symptoms, that kind of wasting, melting. However, it didn't reach this large scale. It kind of happened very local to regionally and didn't affect the entire West Coast. So that raises such a host of questions, like what, you know, is it the same pathogen? Has the pathogen changed? Was the pathogen weaponized by these high temperatures? Like, how did it happen so quickly across the entire coastline? I'm trying to remember, how did they rule out the densovirus? I think it had to do, so one of the things that's really tough is that it's really hard to find clean stars. So sunflower stars that we know haven't been exposed to the disease. So that's one of the ways they do this, disease testing, and they try to find the pathogen, is they take clean stars, undiseased stars that have never been exposed to it before. They put them in tanks and they pipe in, bacteria or virus or whatever it is. And because we can't figure out whether these stars are clean or not, the survivors, they may just have some kind of resistance to the pathogen. And so they may not truly be undiseased. And we don't know if juveniles raised in captivity, we don't know how this thing spreads. We don't know if it's in the water. We don't know if they're all born with it, if it's like some kind of bacteria they normally have that just grows out of control. So we thought it was the densovirus. There was some good evidence there, but kind of getting that last level of like definitive evidence that when we add this virus, then we see wasting, it's just really, really tough to do when we don't know what stars already have it.
Thanks. I have one question for you. There's the secondary effects of, say, absent otters or absent sunflower sea stars, the secondary effects being on kelp. What about direct effects to kelp in terms of water temperature? Could you describe sort of the conditions that kelp grows better or less well in terms of the water temperature?
Yep. So kelps are fundamentally cold water species. They need cold, clear water that has lots of nutrients. One of the reasons they like cold water is because they're so fast growing. They need lots of nutrients. These are one of the fastest growing species in the world. Kelp can grow like a foot a day. And so if you don't have like a bunch of nutrients hanging out in the water, they can't grow as quickly. And so cold water has more nutrients. And so they are just fundamentally limited to temperate and polar environments. And so there's no way to get around it. Heat waves threaten kelp. Heat waves and warming ocean temperatures, those are actually two slightly different things. They threaten kelp for the most part because of the way we're not sure yet if it's because warm water holds less nutrients or if it's because of the actual temperature itself. There's a lot of research going into that right now. So climate change is a major threat to kelp forests everywhere. And then additionally, I mentioned that they need clear, uncontaminated water most of the time because they need a lot of light to be able to photosynthesize. And so in places, one of the things they're worried about in the Puget Sound, where they're starting to see declines in their kelp populations, they see more declines in kelp in the Puget Sound near urban centers. So that leads them to think, does this have something to do with water quality? Is there pollution or runoff or something else that's making the water cloudy and making it harder for kelps to grow? So I just say that to point out that that climate change is kind of the big elephant in the room. It's a huge problem and we don't know what to do about it right now. But also there are a lot of other human impacts, things like agricultural runoff, water quality in the terms of like erosion and sedimentation, harvesting. There are a lot of other things that humans do that also stress out kelp. And if we could eliminate some of those, it would just be one less threat for kelp populations to be dealing with as they try to adapt to this new warmer world.
I may have missed this question before, and you can tell me if I'm repeating myself here. But can you speak to the relations between the Pycnopodia declines and other sea stars?
Yeah, that's a great question. So the scientific community has a feeling or has some good evidence that different species had different levels of resistance to this wasting syndrome. And so, for example, when I go out and go diving, when I was diving this weekend in Port Orford, I saw dermisterious sea stars, which are just another species of them. I saw dermisterious sea stars all over the place. I saw Henricia sea stars pretty regularly. And so there are kind of two phases at which we know there can be different responses amongst different species to this disease. One is how resistant are they to the disease at first? And so we know that some hard-bodied sea stars, so some sea stars are kind of squishier and some have more internal structure and are more firm. We know that those harder-bodied firm sea stars tended to have their populations decline less in some places. So they might have been more resistant to the disease itself. And then there's the how quickly can they rebound post-disease. And so things like Pycnopodia, the ochre sea star, it was horribly impacted by wasting, but we're already seeing populations kind of bounce back. Does that mean they evolved some kind of new resistance to the disease, or does that mean they can just reproduce more quickly? Maybe they have shorter lifespans, and so it's easier for them to bounce back. We're not sure yet, but we believe that Pycnopodia was essentially probably the sea star hardest hit initially, and with some of the least recovery since wasting really broke out. Gotcha.
I know that you've mentioned some of these organizations, but could you just give, again, a quick summary to the folks listening, some of the resources folks could pursue outside of the Elakha Alliance, obviously, about sea stars and about kelp conservation?
Yeah, that's a great question. In terms of sea stars, I showed you that email or that web address earlier, seastarwasting.org. There's a lot of good information on there, and they've also been tracking community reports of sea star wasting since basically the beginning of the outbreak. So you can get a lot of good information from seastarwasting.org. I'm trying to think about what are some of the cutting edge groups studying kelp forest health and resiliency. The Nature Conservancy, honestly, in California, really responded to this collapse in Northern California of kelp forests pretty aggressively, and has spent a lot of money trying to support all the managers and community members and scientists saying, well, what do we do now? So the Nature Conservancy has been thinking a lot about that. California Department of Fish and Wildlife, because that collapse in Northern California knocked out their red urchin and their abalone fisheries, California Department of Fish and Wildlife is involved now, and they're putting out some resources. I would also say the Puget Sound Restoration Fund. I'm pretty sure that's their name. They, as I mentioned, they're starting to see declines in kelp in the Puget Sound, and they just recently wrote up a big report about why they think it's happening and what they might be able to do. Great. So off the top of my head, those are the ones that I'm thinking of.
Yeah, that's great. And I've got one more question, final one. You mentioned, we talked about ocean warming, but specifically ocean acidification. Is there any anecdotes or any thoughts about any biochemical pathways that might to do with ocean acidification and the sea star?
Yeah, and the sea stars, oh dear, another thing that I should be better read on. I do know that most sea stars do use some kind of calcium, do have like calcium not skeletons the same way that like sea urchins or corals do, but they do, like for example, the Pycnopodia sea star has what they're called ossicles. They're these little bony bumps around their body that I think act to prevent predators from wanting to eat them because they're not very tasty. And so I think a lot of echinoderms do, and sea stars are a type of echinoderm, do use calcium more than maybe other species. So as far as the adults go, I think ocean acidification probably has the potential to be impacting them. Additionally, I am not an expert on the larval phase, so the really tiny juvenile phase of Pycnopodia. And oftentimes juveniles we're finding are some of the ones that are being hardest hit by ocean acidification. So I wish I had better information for you.
No, that's great. Thank you for surviving this long Q&A session, but that's great.
Yeah, I'm glad we got to a lot of questions. These were great. And I appreciate hearing what everybody's wondering about.
Well, thanks again, Sarah. And I'll pass it over to Bob.
Okay, I think I'm unmuted here. Am I unmuted? I think so. Well, thanks everybody for hanging in here. Let's take a 5-minute break, allow new people to come in. And once again, a big round of applause for Sara. That was great. That was exactly the kind of thing I was hoping you'd say. So we'll see you all in about 5 minutes at the start of the hour. Thanks.